So the next part here we come to the alteration of generations. Let us just take a look at the life cycle of Edogonium. Now this is the life cycle in case of Macarandrus monoecious, meaning in Macarandrus species where both the male and the female structures, they are present on the same, they are born on the same filamen, so they are monoecious. Okay, so in case of Macarandrus monoecious, you can see the sexual reproduction is the same, where from the Edogonium filamen one of the cell will act as a zoosporangium, and inside the zoosporangium the zoospores will form. Then these zoospores, under favorable condition, they will germinate and give rise to a mature filament. But in case of sexual reproduction, we have, in case of monoecious, both the Oedogonium and the Antheridium, they are born on the same filament. This is the diploid filament will give rise to, this monoecious, Macarandrus monoecious, we know that the zoospore will give rise to a haploid filament. Okay, so from here we get a haploid Oedogonium and a haploid Antheridium, which will give rise to the haploid egg and the haploid antherozoid respectively, the male and the female gamete. When these male and female gamete, they undergo fertilization, because haploid and haploid, they combine, okay, during fertilization they will fuse together to give rise to a diploid zygote. Now X and X becomes 2X, right? A diploid zygote. This zygote, we know that it will undergo meiosis to give rise to the haploid zoospores. Now from diploid we are getting the haploid zoospore, and this will germinate to give rise to a haploid filament. So from a haploid zoospore we get a haploid filament. Now in case of Macarandrus dioecious, where the male and the female filamen, they are born on different, the male and the female Antheridium and Oedogonium, they are born on different filament, that is dioecious is the same thing as you can see. Okay, from a filament, a male filament which is haploid, we get antherozoid which are haploid, and from a female filament which is haploid, we get the egg which are haploid. When these they fuse together during fertilization, they will give rise to a diploid zygote, which will undergo meiosis to give rise to haploid zoospores, and these zoospores on germination, they will give rise to both haploid male and haploid female filament. Okay, so now this one, it shows the life cycle of the Nanandra species, and in Nanandras we know that both these filaments, they are always born on, the male and the female structures, they are born on different filaments. That is why it is always dioecious Nanandras. So the asexual reproduction is the same thing. From a filament we will get a zoosporangium, and inside the zoosporangium the zoospores will form, which upon germination they will give rise to a mature Oedogonium filament. And during sexual reproduction, it's the same thing as in the case of Macarandrus species, where the male filament, it will give rise first to the androsporangium. Remember, in case of Nanandrus, because we have dwarf male, so first it will not give rise directly to the Antheridium. Just remember this. The filament, the male filament, it will forms the androsporangium, which will give rise to the androspore. And upon germination, this androspore, it will form the dwarf male. Okay, then in the dwarf male we will get the male structure, okay, the sexual structure of male, that is the Antheridium, which will give rise to the male gametes. And in case of female filament, it's the same thing as in Macarandrus, where we get the filament will act as the Oedogonium and will give rise to the egg nucleus, that is the female gamete, and when these two haploid spores they fuse together during fertilization, they will give rise to a diploid zygote. And again, they will undergo meiosis to give rise to two haploid zoospores, one from the male and one from the female. So these zoospores, upon germination, they will give rise to the male and the female filamen. So you can see in alteration of germination of generations in case of Edogonium, the diploid stage here is short-lived. You can see only in this part there is a diploid stage, right? So it is short-lived. It is found only in the zygote. The diploid stage here is found only in the zygote from the fusion of the male and the female. In all these three cases, what do you see? That the diploid stage is found only in the zygote, right? Nowhere else you can find only the haploid stage, but only in this stage we can find the diploid. So it is short-lived. And the diploid nucleus of the zygote, it will form 4 haploid zoospores again. Okay, it will give rise to the haploid zoospores again by meiotic division. And these haploid, we know that upon germination they will give rise to haploid filament. So therefore you can see that the life cycle of Edogonium is haploid predominant type. Okay, remember this, that the life cycle of Edogonium in all these three cases, you can see it is haploid predominant type, because the diploid stage is short-lived. It is found only in the formation of zygote after fertilization of antherozoid and egg nucleus. So the haploid stage is predominant type.